Hi, I'm Sharon Long from Stanford University, and I'm here to give my second lecture on nitrogen-fixing symbiosis. Today I'll be talking about the regulation of the genes in the bacterium itself and how those genes are controlled. So just as a review, nitrogen fixation happens in root nodules. These nodules form as the result of action of soil bacteria interacting with a specific plant. Here you see nodules growing on the root system of a soybean. This is a complicated series of tissue and cell interactions, a complex developmental process, and there's specificity between the bacteria and the plant. I'll talk about one side of that specificity a little later today. In the early stages for sure, and I'll make the case that it's in the later stages as well, bacteria and plant exchange signals in order to coordinate with each other. Now, let's take the standpoint of the bacterium. It's making this huge transition from the free-living bacteria through invasion, in which the bacteria are able to form an infection thread and penetrate through individual cells, and then into multiple layers of the root, and finally nitrogen fixation, where the bacteria have become endosymbionts inside the cytoplasm of a plant cell, and they persist there in a differentiated form, almost like an organelle. All of these changes are accompanied by carefully regulated changes in gene expression. So here's an outline of what I'd like to tell you today. I'll begin by talking about a few tools that we use to study the genetics and the gene expression of the bacteria. And then I'd like to say something about the control of the nodulation genes. This is probably the most studied of any of the gene regulation events in symbiosis. I'll review some of the background, and then I'll talk primarily about the protein activator Nadi. I'll then tell you some of our recent work on RNA polymerase, and especially the sigma factors, and how they may be playing a role in changing and guiding gene expression during symbiosis. I'll finish with several glimpses of how particular sets of genes are controlled, and a preview of all of the factors yet to find in terms of how many genes are active in nodulation. So the first tool to remind you about is my favorite, and that's genetics. We can take bacteria, and through mutagenesis and then blind screens, we can ask what are the most important genes for symbiosis. Now, there's a couple of different symbiotic phenotypes that we have observed. In contrast to a wild-type situation where the bacteria growing in the free-living state are able to interact with the correct host plant and make functional nodules, shown here by having a red color reflecting the leg hemoglobin that's inside the plant. The nodules harbor bacteria that fix nitrogen. The plants are able to use that to make chlorophyll and protein. They flourish and they're nice and green. But if the bacteria have a mutation in a gene that's required for the formation of the nodule, the result will be that the plant forms no nodules, therefore it's not getting any fixed nitrogen, and it starves. It's yellow, and it doesn't grow very well. We would call this a nod gene in the bacterium because the mutations in that gene result in the so-called nod minus, no nodule phenotype. The other outcome, again comparing to the wild type, would be that a bacterium might be able to form a nodule, but those nodules don't function. And what I've shown here is I've symbolized that by these empty white circles. Those show nodules which are not fixing nitrogen. They don't have leg hemoglobin. And so we would call this fix minus phenotype, and the corresponding bacterial gene is going to be a fixed gene. Another approach is to look at the genome. Bacteria tend to have genomes between about 1 million for the very smallest pathogens, all the way up to 10 million bases. Rhizobium is on the large size, over 6.5 million bases, divided into 3 replication units. The chromosome, 3.65 million, and 2 very large so-called megaplasmids. They're called megaplasmids because they're more than 1 million bases each. PSYMB, 1.68 million, and PSYM-A, 1.35 million. The nodulation and nitrogen fixation genes are located on PSYM-A, but in addition, there are symbiosis genes on PSYM-B and on the chromosome. So to some extent, the symbiosis genes are dispersed. Now, we can take a genomic approach to look at transcription, and one way in which we have approached that is to do global assays of transcription using a custom-designed Affymetrix chip. On this chip, we place the genome of the bacterium together with 10,000 some genes from the plant as follows. The entire Rhizobium mellolodi genome is there, every one of the 6,000 plus open reading frames, and the intergenic spaces, and 10,000 tentative consensus sequences from cDNAs that were sequenced from the plant. Now you'll see some experiments later in which we're looking at the expression of genes in Sinorhizobium mellolodi, and in one case we're going to be simultaneously looking at both the bacterium and the plant. And we can do that from a single RNA prep as shown here. So if you grind up a nodule, so you're starting with the nodule over here. Of course, this is going to be full of many plant cells. Each one of those plant cells has bacteria, and those bacteria shown here have differentiated. So a nodule lysate is going to have a mixture of all of those materials. We have a method which allows us to capture both bacteria and plant DNAs at the same time, and that can be shown here. For reference, in the right-hand lane, we have isolated RNA from a plant root. That shows you the large and small ribosomal RNAs here for reference. On the left-hand lane, we have bacteria grown in culture. You can see, again, the bacterial RNAs here. Our prep for nodule RNAs captures both of those, as you can see. And this, by being labeled and hybridized onto our Affy chip, allows us to get a snapshot of both partners at the same time, which is very useful, especially when you're analyzing mutants, and useful because perfect synchrony of nodulation is hard to achieve experimentally. So, having introduced those tools, now I'd like to tell you about the control of the nodulation genes. Just a little bit of review. Nodulation genes here are the genes that encode enzymes that synthesize nod factor. I talked about nod factor, and I'll show you a cartoon in a moment. It is the substance created by the bacteria. That's a molecular signal that causes the plant to form the nodule, and it also causes transcription and a number of other events in the plant. So, powerful signal molecule. The genes in the bacterium that are responsible for producing nod factor include the common nod genes, nod ABC. The transcription of nod ABC is controlled by a promoter that includes what's shown here in this oblong, the so-called nod box, a highly conserved motif that's in the DNA of upstream of the nodulation genes, both this set of common nod genes and others that are coordinately regulated. All of these genes, as I mentioned before, are located on PSIM-A. Now, this cartoon introduces a couple of themes. First, it reminds us from my first lecture that the early events of nodulation include a conversation between the plant and the bacteria. The plant sends a signal out to the bacteria send a signal back. In the case shown on the left, we're taking a look at alfalfa, Medicago sativa. This plant produces a molecule coated in yellow here, and you'll see the structure in a moment. That signal from the plant to the bacterium is able to trigger gene expression, and we'll talk about that in detail. The result of that gene expression, as I mentioned before, is the production of a nod factor, called here NFSM. That's the nod factor from Sinorhizobium mellolodi. It has a backbone of N-acetylglucosamines, which is modified in the case of Sinorhizobium mellolodi with a 16-carbon N-acyl substitution here, an acetyl and a sulfate, each shown by a color code. Now, on the right, we have the contrasting example of a cousin, a related legume, pea, or Pisum sativum. Here we've got the root of the pea plant. It's creating a flavonoid signal whose structure is slightly different, and I've shown that just by the color coding. That signal triggers Rhizobium leguminosarum, which is the symbiont for pea, to transcribe its nodulation genes. And those encode enzymes that make a nod factor, but it's a slightly different nod factor. The primary difference that I'm showing here, which is not the only difference, but for the sake of simplicity, a primary difference is the nature of the N-acyl substitution. It's 8 carbons instead of 16, and it has 4 unsaturations. So what we see is some specificity. A signal that's specific, that goes out from the plant, from alfalfa here, or a slightly different one from P, and the corresponding bacteria which respond to this signal make a slightly different nod factor as shown here. So this conversation, plant to bacteria and back again, already has some of the hallmarks of the host specificity. We're going to talk only about the first part of that conversation. Here I'm showing again the host plant producing a flavonoid signal, and that is causing transcription in the bacteria. Now I'd like to focus in on the interaction of the flavonoid with a protein encoded by this gene, Nadi. Nadi is an activator protein, a part of the lysR family. Now, what are all of the elements we're going to have to look at? Of course, at a promoter, there's going to be an RNA polymerase. There's going to be a DNA motif to interact with. In this case, there's an activator. And we wonder, how do these plant signals interact with all of these components in order to achieve transcription? And how does this occur in a specific way? So I'll take a look now first and most specifically at the protein Nadi. I'll put that here in the context of all of those other components. The red here is showing DNA. All right, so here's the DNA, and what the object of this regulation is going to be to achieve transcription of the nodulation genes so that they can then make the proteins that synthesize nod factor. At the DNA, we have a promoter, and that's upstream of the nod boxes. We know that Nadi binds to the promoter and bends the DNA. I've shown here a model of Nadi as a tetramer. We don't know exactly what its form is, but we suspect that it's most likely to be a tetramer, and more work in the future we hope will elucidate that more precisely. We've shown through genetics that the ability of Nadi to cause gene expression in bacteria in response to a plant signal requires the chaperonins, GRO-ES and GRO-EL. Also, we found through genetics that this entire system requires the stringent response, that being a response classically to starvation, where a protein here, REL-A, causes the synthesis of guanosine tetra or pentaphosphate, shown here as this star. All of that has to interact with RNA polymerase, the machine that's going to interact with DNA and cause template-driven transcription of an mRNA off of the DNA template. So these are some of the things that we know, but there's a lot of unknowns. Let's ask, for example, how the inducer works with the Nadi. Now, I'm not going to show you the data on our GRO-EL work, but I will just mention that we have a combination of genetic data and in vitro biochemical studies that show how GRO-EL is required for Nadi and luteolin to be active. In one of the experiments that follows, I'll be referring to that some more. Let's now just ask about flavonoids, including the particular flavonoid luteolin and Nadi. So in this experiment, we're doing the following. We're growing rhizobium either without an inducer or with an inducer. So it either has just culture medium or it has the plant signal in it. From those cultured cells, we purify Nadi, and we ask, using electrophoretic mobility shift assay, does that Nadi bind to the nod box promoter or not? So in pairs here, you're going to be seeing in the open boxes how much DNA binding is accomplished by this amount of Nadi onto the nod box promoter, the nodulation gene's upstream region. And this is the amount of binding when the Nadi in question came from cells grown without inducer. But you can see here that if the Nadi had been purified from cells grown with inducer, the same amount of Nadi does much more DNA binding. So it appears that the affinity of Nadi for its DNA target is increased if the Nadi was purified from a cell exposed to the plant inducer, luteolin. Now, this is an in vitro study. In this case, all of the Nadi is being purified from cells grown with no inducer. That purified Nadi is then incubated with buffer only or with the plant inducer luteolin. So we're going to have minus for no luteolin and plus for added flavonoid luteolin. We found early on, in data not shown here, that if this were done with Nadi only, there was no result. However, if we carried this experiment out in a system where the Nadi was also presented with GRO-EL, GRO-ES, magnesium ATP, and inducer, there was a profound effect. And that's shown here. The primary data are as follows. In each case, we've got purified Nadi that was incubated just with magnesium ATP and GRO-ES. Here, GRO-ES, GRO-EL, or with those same components and luteolin. Just chaperonins and ATP here, or with luteolin. Components only, or with luteolin, and so forth. And what you can see is that the presence of the luteolin, the flavonoid inducer, is able to enhance the ability of purified Nadi to bind to its target DNA sequence in the nodulation promoter. So what we've got here then is a demonstration that in the presence of chaperonins and in the presence of the flavonoid inducer, again we see enhanced ability of Nadi to bind to DNA. And that's quantified here. If you look at the fraction bound, absence of luteolin in the incubation mixture, presence of luteolin. Now, this was exciting to us because we were starting to get at the question of how the flavonoids affect the bacteria's ability to express genes. But recall, I told you that one of the hallmarks of this system is that it is specific. We know that different plants make different flavonoids. Some of them work in vivo and some don't. Is that due to DNA? Knowing that we should expect any in vitro regulatory system to explain specificity as well as other aspects of Nadi activity, we carried out an in vivo experiment based on some earlier data from some years ago that showed a correlation between what species a nodulation Nadi protein came from and what kinds of flavonoids a cell would respond to. So in this experiment, genetically, we've got a responder cell that's going to report for us whether nodulation genes are transcribed or not. It's got a Nod gene promoter, fused to beta-galactosidase. But that genetic construct has its own Nadi taken away. There's a deletion for Nadi, but there's a Nod-lac fusion. Into that strain, we're going to place one or the other of these two plasmids. These plasmids have the same vector, so the same backbone. They also have a promoter that's exactly the same. It's a constitutive promoter that's highly expressed. What's different in them is that in this strain, we've got a gene that represents the trimmed open reading frame for Nadi from Sinorhizobium mellilodi, which is the symbiont of alfalfa. Now, in this other plasmid, we have the trimmed open reading frame of Nadi from Rhizobium-legume trifolii, which is the symbiont of clover. So everything is the same except the actual amino acid sequence of the two Nadis. Now we're going to place those into the reporter strain that has no Nadi of its own and ask whether Nod genes are expressed or not. So in the left-hand column here, what you're seeing is whether or not Nod genes are expressed in response to luteolin. And what you can see is that when the cell includes this plasmid that has the Sinorhizobium mellilodi Nadi, then yes, there is Nod gene expression. However, if the strain has the Nadi gene that came from Rhizobium trifolii, then it's with kind of iffy. There's not very much expression. So this suggests that this Nadi can induce in response to this molecule, but this Nadi cannot induce very well. But now let's ask, how does those reporter strains respond to a completely different flavonoid shown here? And you can see, for example, it has only 1 hydroxyl, whereas luteolin has 4. Well, now, if we take the strain containing the Rhizobium elulodi Nadi, we see that there's no expression at all, whereas the Nadi from the Rhizobium trifolii shows good activity. So this shows that the amino acid sequence of just Nadi is sufficient to account for a different response to one flavonoid structure versus another flavonoid structure. We wanted to put that together with the data that I just showed you that demonstrated that Nadi can bind better to its NOD-box target if it's got luteolin present. But what we found was something of a surprise. Now, what I'm showing here on the top are the structures of the flavonoid I've introduced before, that's luteolin, and a set of flavonoids here that in vivo do not cause gene expression in Sinorhizobium elulodi. So we would infer from that that the Nadi of Sinorhizobium elulodi cannot interact productively with any of these because they don't work in vivo. However, if we carried out experiments similar to those I showed you before with each of these, we got the following. Here is a set of data, and here's another. This for daidzein, that's the molecule up here. This one for areodictyol, which is the molecule there. And what you can see here is that if you compare Nadi protein incubated with components such as GRO-EL and magnesium ATP, or those same components plus the flavonoid in question, these flavonoids, even though they don't work in vivo, they still enhance the binding of Nadi to its target promoter. That's true for daidzein, it's true for areodictyol, and what this shows is that DNA binding is caused even from flavonoids that are not active. And some of the examples of the data and a quantification are shown here. This shows the gel shift of Nadi from Sinorhizobium elulodi presented with buffer only, with daidzein, luteolin, and naringenin, 3 inducers, of which only the luteolin is really active in vivo. Nonetheless, all of them cause gel shifting, and that's quantified here. What you can see in this graph is DNA, Nadi with no inducer of any kind added, and Nadi with the 3 different inducers, some of which are active in vivo and some of which aren't. So right now, our working model is that Nadi is able to bind DNA with an increased affinity no matter what flavonoid is there. And in fact, the flavonoids that do not work in vivo may actually bind to Nadi and inhibit the ability of the correct flavonoid to induce by causing unproductive DNA binding. So DNA binding does not explain the specificity of Nadi for the flavonoid inducers that come from different plants. That leaves us with a series of new experiments to do in the future, and among them, what we want to ask is, does the presence of the correct flavonoid somehow affect the interaction of Nadi with RNA polymerase? And that's led us in turn to ask about the components of RNA polymerase, and in the end, we hope also to work in vitro with the guanosine tetraphosphate and ask about all of these components together. But in this next section, I'll just like to talk about RNA polymerase. In particular, I want to talk about the sigma factors. Now, just as a review, RNA polymerase in prokaryotes has a core made of 2 alpha subunits, 2 copies of the alpha subunit, a beta and a beta prime subunit, and in addition, a sigma factor, which has multiple domains, and they're shown here in this cartoon, which is derived from E. coli sigma factor. And what we now know from work in many labs over the years is that there are many kinds of sigma factors. Originally, the one that was best characterized was the housekeeping or RPOD sigma 70 type of sigma factor. And that's the form of sigma factor that reconstitutes with RNA polymerase for the transcription of most genes in E. coli and in other organisms. The sigma factor here has various alpha helices and other subdomains. That's what these letters are for. But this is one polypeptide as it interacts with different parts of the beta, prime, beta, and alpha subunits. But in addition to the RPOD, there are other sigma factors. A stress sigma factor, RPOS, which is very prominent in survival responses of E. coli, heat shock, RPOH. In some enterics, there's also RPON, which is important for nitrogen metabolism and for other functions, such as nitrogen fixation in Klebsiella, and a number of other sigma factors. And I'm going to come back in a few minutes to one in particular, the RPOE. In Rhizobia mellilodi, our annotation showed that there's at least double that number of sigma factors. One of the striking differences with enterics is that there's no copy of RPOS. However, there is a housekeeping sigma. It looks very comparable to the sigma 70 of E. coli. There are 2 copies of the RPOH, or so-called heat shock sigma. There are 10 copies at least of RPOE, in addition to the FEC-I, originally identified for relationship to iron control, and the RPON that's important for nitrogen fixation. In the following section, I'd like to tell you some of our new work on RPOH and refer briefly to some emerging work on RPOE. We wanted to know what do those 2 different copies of RPOH do? Are they important for heat shock? Are they important for symbiosis? Let's start with a look at how these are expressed. On the left-hand column, you can see how the 2 different copies, RPOH1 and RPOH2, are expressed in free-living cells. The RPOH1 tends to be expressed mostly in late log, late exponential, and stationary, but RPOH2 is expressed mostly in stationary cells. If you grind up nodules and ask whether these RPOH genes are transcribed, you can see that RPOH1 is transcribed. Yes, it's present in nodules, but RPOH2 was not detectable in our assays. That leads to a surprise in a minute, as I'll tell you. Knocking out one or the other or both of these RPOH genes leads to some phenotypes in the free-living state. The RPOH1 mutant is very sensitive to high temperature. RPOH2 does not appear to have much of a phenotype, but the double mutant, similar to the RPOH1, is affected by high temperature. Here's the surprise about the symbiosis. What we found was that a mutation in RPOH1 was fix minus. RPOH2, if it's knocked out only by itself, does not appear to have any symbiotic defect. However, if you knock out both RPOH1 and RPOH2, and despite the fact that RPOH2 doesn't seem to be detectable as a transcript in nodules, remarkably, that's nod minus. That means that one or the other of those RPOH sigma factors has to be functioning in order for the symbiosis to proceed past some of the very earliest stages. So that's a mystery that we are beginning to look at in more detail. Meanwhile, we've also asked about the spectrum of genes that are expressed. In the following experiment, we used the Affymetrix chip that I described earlier. And what we're going to be doing here is taking a look at the transcripts in wild-type cells and in each of the 2 single mutants and the double RPOH1H2 double mutant as we grow them in culture, give them a heat shock, and then harvest the RNA to assess global transcription. Now, in the following, I'll show you some summaries of our data. Remarkably, we found that when you heat shock wild-type Rhizobia, almost 1/3 of the genes in the genome, over 2,000 genes, are affected one way or another. 1,000 of them go up, another 1,000 go down. Another intriguing feature of this transcription picture is shown here. Now, if you take the entire genome of Rhizobium and you look at the distribution of open reading frames, you can see that more than half of the open reading frames are in the chromosome, and that's shown in this here. So that's the chromosome. PSYMB accounts for this many as a fraction of the whole, and this is the number that are in PSYM-A. That's the total genome annotation. But if you look at those 2,000 genes that are changed one way or the other by heat shock, what you see is that the chromosome is underrepresented and the two symplasmids are overrepresented. So that in and of itself was kind of intriguing, especially when you put it together with the fact that the double mutant of both those sigma factors is very defective in symbiosis. Here was another surprise. If you take a look in this picture only at the genes that are increased in heat-shocked cells, and that's, as I said, over 1,000, what we find is that the large majority of those are increased by heat shock even if the two RPOH sigma factors are both knocked out. And that's shown here. These are the increased genes by heat shock in a wild type. And this blue circle shows those genes that do not get enhanced in a mutant. Right? These are the genes that do not get enhanced by heat shock in an RPOH1, H2 double mutant. And the overlap is only about 200. So only a minority of the heat shock genes are apparently controlled by the RPOH sigma factors. So they're doing other things, and some other regulatory system is taking the job that in E. coli would be done just by RPOH. Another surprise that needs to be followed up. So I'd now like to ask the question, how about the other sigma factors? As we move forward looking at RPOH, we know that there are more sigma factors to account for in Sinorhizobium eluloti. Now, one that's important for sure that I won't say more about is obviously the RPON sigma 54 sigma factor, known to be required for the transcription of nitrogenase and for a number of the other genes in the late differentiated state. The RPOE family presents some other very intriguing puzzles. Now, RPOE was identified in E. coli as a sigma factor responsible for genes that were triggered by denaturation of proteins in the periplasm. They are controlled post-transcriptionally by an anti-sigma factor, which captures them in an inactive form unless the anti-sigma factor is somehow triggered to release the RPOE sigma, which can then go and associate with core polymerase and turn on a large number of genes. So we've begun to look genetically at these RPOE genes. We found that the single mutants in each of the RPOE open reading frames don't have any particular phenotype, and that's shown here. Right? Single RPOEs have some slight membrane sensitivity, but basically they seem normal in culture and they're normal for symbiosis. We've now also gone through and made all of the double mutants for each of those sigma factors. What we find there is that some of these RPOE sigma factor double mutants have a symbiotic defect. They make nodules, but the nodules do not fix nitrogen. That suggests that somewhere in the transition between the beginning of nodule formation and the differentiation of the bacteria, essential genes require the activity of some of the RPOE sigma factors. It also points out the likelihood that these are at least partially redundant. That's why none of the single mutants had a phenotype, but some of the doubles do. So in our future work, we are now asking which of the genes are controlled by each of these RPOEs, and can we use transcriptional analysis of the global picture of RPOE control to understand the redundancy and understand where they're required in symbiosis. So from that view of the RNA polymerase, I'd like to go to a few more case histories, but we need to keep in mind that whatever stages we're looking at, we may need to consider the global picture of RNA polymerase and of other regulatory contexts to understand how specific regulatory circuits are able to function in symbiosis. Now, my next topic is about the control of extracellular polysaccharides. EPS, extracellular polysaccharides, are required for symbiosis. And I'll tell you in this section about how these are regulated by a family that includes a periplasmic regulator and a two-component regulatory system. Now, the EPS in Sinorhizobium mellilodi has been studied in detail by Graham Walker and his colleagues at MIT over a number of years. The name of this exopolysaccharide is succinoglycan because it is modified by acidic groups, including succinyl groups. And they have shown that mutants which are not able to synthesize succinoglycan are able to provoke nodules to form on alfalfa as a host, but they cannot invade. So what's shown here, which is a root hair with bacteria invading, and these bacteria are labeled with green fluorescent protein, allowing you to follow their invasion into the plant. This process will not occur unless you have the extracellular polysaccharides. Now here's some mutants that were found by Graham Walker and his colleagues that were able to make polysaccharide, but they don't make the right amount. In fact, they make too much. So what you're seeing here are bacteria that are streaked on a solid medium, including a dye called chalcofluor. Chalcofluor in solution is not fluorescent, but when it binds to certain beta-glucans, it becomes fluorescent. It's used sometimes as a laundry brightener because, among other glucans, chalcofluor can bind to cellulose, which is the constituent of cotton, and when it binds and becomes fluorescent, that means that any cotton fabric that has chalcofluor will fluoresce slightly in sunlight because there's a little bit of UV in sunlight, picks up the UV, fluoresces bright blue. That makes the laundry look whiter than white. Now in the case of rhizobium, it's lucky for us that the succinoglycan also binds chalcofluor. So a wild type, which you can see here, has a light white glow when it's grown on chalcofluor medium and exposed to UV light. But two mutants, here an XOR mutant that's defective, and here an XOS mutant that's constitutively active, that it's hypermorphic. Each of these makes too much polysaccharide. And actually, it turns out that's not good for symbiosis either. If a bacteria makes too much symbiosis, it doesn't do very well at invasion. So what's going on here? Why does this mutant make too much polysaccharide when it's overexpressed, and this mutant makes too much polysaccharide when it's defective? Well, we would guess that that's because this is a negative regulator and this is a positive regulator. But we took these and found fortuitously some unexpected phenotypes that went beyond polysaccharide. Here's the first of those, and that is that mutants defective in XOR or overproductive of overactive in XOS are not motile. They are not able to swarm on a motility plate, and that's shown here. This is a wild type. You can see that on this particular auger, it's able to swim around. It makes a big, wide colony. But the XOR minus mutant or the XOS overexpression mutant, both of these are not able to swarm. That's because they're not motile. This plate also lets me introduce to you two genetic constructs that give us clues. In the first one over here, we've taken this XOS mutant and we've taken a wild type XOR. That's the inhibitor, what we infer to be an inhibitory factor. We've taken XOR and we're overexpressing it. If we overexpress that XOR in the presence of this overexpressed XOS, it apparently allows the cells to be normal again. So that suggests to us that XOR actually inhibits XOS. And even if you have a hyperactive XOS, if you then add more and more XOR, you're able to get things back to the wild type balance. Now the second clue was also very interesting. Here's XOR, and in a population of XOR mutants, which are, as you can see, non-motile, a suppressor arose. That suppressor allows the XOR to be motile again. So when we tracked down the gene and sequenced it, we found that that suppressor was actually in a gene called CHVI. CHVI is a DNA binding protein that's linked to XOS, which is a response regulator. XOS and CHVI are a two-component system. And without even asking about XOS, what we did was to mutate XOR and find a suppressor that was in the substrate of XOS. Now, motility appeared to show us a linkage of the XOS and the XOR with CHVI. But how about other phenotypes? Well, one of the other dramatic changes that happens in an XOR minus or XOS minus XOS constitutive bacterium is that genes which are normally upregulated in a wild type are very low in expression in those mutants. And you can see that here. If you take a look here, these blue panels, these are actually two duplicates of an XOR minus and then an XOS minus, showing a whole set of genes that are not appropriately regulated if XOS and XOR are in those defective conditions. The two suppressors that I showed you, however, the ones that reverse motility, they also reverse the transcription defects and restore them to wild-type levels. And that's shown in these panels on the right, where you can see that coordinate restoration of a wild-type function from these same suppressors. So what we find is that the motility defect and in the transcription profiles, XOS and XOR and CHVI all appear to be targeting coordinated functions. We believe, based on that on some biochemical data, that the system works as follows. We've shown biochemically that XOR is in the periplasm, in between the cytoplasmic membrane and the outer membrane. So as a negative regulator that's present in the periplasm, it's unique at this point. There's no model for how this might be working because there's no other proteins of that type yet reported. XOS and CHVI are the two-component system with XOS as a histidine kinase and CHVI is the response regulator that binds to DNA. And through CHVI, all of these functions, including motility, transcription, and we've also shown biofilm formation and other functions, they're all controlled. Now, this presents us with a lot of mysteries. For example, kinases, sensor kinases, are presumably responding to some kind of change in the environment, some signal. What is it? Is the plant sending something? Are other bacteria sending something? What are the factors in the environment that control the activity of XOS? XOR as well. How does that act? Does that also respond to the environment? So these are some of the mysteries that we hope to work on at the molecular level. And one of the other directions we're going right now is to look very specifically for the targets of the CHVI DNA binding protein. Now, XOR and XOS, as I showed you, control a lot of genes. There are more genes out there. Now, we already know that this regulon is required for exopolysaccharide. It has to be just right in order to get invasion. You saw that there are many genes that are controlled. But we also think that as you go from invasion all the way to differentiation, there are many more genes than that. First, in a promoter trap screen, genetically, we found several dozen bacterial promoters that appear to turn on in between the beginning of invasion and the final differentiation. So we're looking at those genes to ask, what is it that controls them? Furthermore, we've recently carried out more genetic screens of the type that I described in the beginning, putting random bacterial mutants on plants and asking whether those bacteria are able to cause nodules to form and whether those nodules are functional, to find more nodulation mutants and fixation mutants. We have already found several new genes not previously recovered in earlier screens for transporters, for example, which suggests that the bacteria must be able to import or perhaps export materials in order to accomplish nitrogen fixation and differentiation. And in addition, we've taken the transcriptional approach as follows. So based on our Affymetrix transcription studies, we were able to show that hundreds of bacterial genes change if you compare cultured bacteria to bacteria in nodules. Over 300 genes increase in expression, including the obvious suspects such as nitrogenase and fixation genes. But many more than that, 900 some genes, go down. These decreased genes include biosynthetic functions such as amino acid synthesis and others. So the bacteria is having a major change in its gene expression profile. The plant changes as well. Now, several hundred genes change, of which 250 or so increase in the expression, and many plant genes decrease in their expression as well. Now, what we wanted to do was ask something about the strategy of how the plants and the bacteria control their transcription in these nodules. So what we're going to do in the next set of experiments is compare the transcripts in a wild-type nodule, such as this one here, to a nodule that's being established on a wild-type plant by a bacterium that's mutated. Now, what mutant are we going to use? We're going to use a mutant called FixJ. Now, the protein FixJ is required for bacteria in the nodule to transcribe the nitrogenase or NIF genes. And that works as follows. Leghemoglobin, which is present in the plant cytoplasm, shown in this cartoon, buffers oxygen so that the free oxygen tension is very low, although oxygen is delivered at a high rate by leghemoglobin. Now, the low oxygen is able to interact with the two-component regulatory system, FixL and FixJ, that activates a signal transduction cascade that results in nitrogen fixation. So here's the plant cytoplasm and the bacterium responding to the plant's signal. If you have no FixJ in the bacterium, then there's no nitrogen fixation. So the bacterium provokes a nodule, it gets into the plant, but then it doesn't work. So the RNA transcription studies we're doing now are going to compare a nodule made with wild-type bacteria and a nodule that's made and infected with bacteria that then don't fix nitrogen. And here are the results. Let's take a look at the wild-type first. As I mentioned, there's and by the way, we're looking only at the transcripts that are going to go up. The plant has about 250 or so transcripts that are upregulated in a nodule. The bacterium has somewhat over 300 nodule transcripts that increase. This is the wild type. Now, if we ask, what happens if we look at the transcripts made in a nodule that was created by the mutant? We see that a nodule made by the FixJ mutant upregulates very few transcripts. The vast majority of these 300 some transcripts that are increased in bacteria when they're wild type apparently requires the FixJ system. So I would say from this that the bacterial transcription profile is responding to some signals right in time at the point that nitrogen fixation is occurring. We found a contrasting result when we ask these same nodules that have so few bacterial transcripts, how many plant transcripts do they have? And that's shown here. What you can see is that almost all of the wild-type transcripts, shown in this outer box, are also made in the nodule that resulted from the bacteria that is mutated for nitrogen fixation. So I would say that suggests a couple of things. First, that the plant isn't making its decision about whether to transcribe these genes at the last minute. It made the decision to upregulate these genes early on before the bacterium makes the decision about whether or not to fix nitrogen, right? So most of these genes, I would guess, are being made to build the nodule in the first place. That the strategy of the plant is to decide on the morphogenesis. But it also shows that, yes, there are some genes, and they're shown in this outer part that's not covered by the overlap. There are some genes that do depend on bacterial nitrogen fixation in order for the plant to express them. So we want to find out in future studies how those are controlled, as well as how some of these are controlled irrespective of the bacterial signaling. And that brings me to the final topic, in which we've been able to use a combination of bacterial genetics and plant genetics as a way of elucidating new signals. In a screen of plant mutants, we asked, can we find plant mutants that fail to accomplish nitrogen fixation, fail to accomplish correct symbiosis? Just as a baseline, let's take a look here at wild type. Now, in a wild type plant with wild type bacteria, you get nice nodules. Here's those nodules. If you do a section through a nodule, you can see all of the infected cells. So here in the corner is the vasculature of the plant, so the root is going like this. Here's the nodule coming out, and all of these dark stained cells are packed full of successful bacteria fixing nitrogen. Using a bacterial strain that has a glucuronidase fusion in the NIF genes, but is still wild type, we are able to see that if you take this kind of section of a nodule and you stain it for glucuronidase activity, you get this deep blue stain, and that means that the nitrogen fixation genes are being transcribed. But now let's take a look at a plant mutant. So this plant mutant is called DNF. That stands for defective in nitrogen fixation. If you see here, the bacterial action, wild-type bacteria on this plant yield tiny little white nodules. They're not functioning. They're not fixing nitrogen. A look inside those nodules shows that the bacteria do get into cells. Here's the vasculature again. Here's the root. Here's the nodule. All of these dark staining cells are packed full of bacteria. However, if you put in the indicator for NIF to glucuronidase and you stain for glucuronidase activity, you can see here that the NIF genes, nitrogenase genes, are not being expressed. There's no blue stain. So something about this plant mutant is not allowing nitrogen fixation to occur. So we can say that the bacteria need whatever it is that DNF1 does in order to differentiate and fix nitrogen. Having carried out the crosses and mapping necessary, we have cloned and identified DNF1. We found that DNF1 encodes a plant protein that is a nodule-specific signal peptidase. Now, using this illustration from molecular biology of the cell, I can just go through roughly what we would expect the signal peptidase to be doing. In the plant cytoplasm, you're going to have the endoplasmic reticulum. Some plant proteins are going to be encoded in the nucleus. They're going to be translated such that they go into the lumen of the endoplasmic reticulum, and that is made possible by a signal peptide. The signal peptidase cleaves off the signal peptide as so, releasing the mature polypeptide product. Now, what do those do? I'll get to that in a moment. But what we would just conclude is that there's going to be something about the action of DNF1 that's going to result in the production of a mature protein that might go to the cell wall, or it might go into vesicles. So whatever is being produced by the DNF1 signal peptidase may be purposed for secretion or for vesicle delivery. So what are the substrates? And here our work was able to intersect with the work of Eva Konda-Roshi and Peter Mergert and their colleagues. They had discovered a family of nodule-specific plant proteins that they called NCR, nodule-specific cysteine-rich proteins. Using our mutant, they have been able to show that these NCR proteins are actually processed by our DNF1 signal peptidase. Therefore, what we can envision is that the lumen of the endoplasmic reticulum, as a donor compartment, is going to be full of these NCR peptides after they're processed, and these may be then delivered to a target compartment. We might envision that as having the rhizobium. So once again, what we can consider here is that we've got NCR proteins from the plant being processed into the endoplasmic reticulum, put into vesicles, and then delivered into a compartment where they actually go into the rhizobium and cause those rhizobium to differentiate. This model can be tested in a number of ways, and those are underway at present. What we can envision, though, is the following. We know that these bacterial cells, which are here shown in a close-up of an infected cell in a nodule, these bacterial cells must be getting a lot of signals from the plant. So if we envision this as a cartoon of a plant cell, and here's the cytoplasm of the plant cell, we already know from previous work that the leg hemoglobin that I mentioned earlier, that gives the nodule its pink color, helps to buffer oxygen. It keeps free oxygen low while delivering it at a fast rate. And we know from earlier work that there's a signal transduction cascade that triggers the activity of NIF-A and eventual transcription of nitrogenase due to the fix-L, fix-J oxygen-sensitive two-component system. But now what we know is that something else is needed. Unless you've got DNF1 processing the NCRs and maybe doing other jobs as well, unless you've got DNF1, you still won't get transcription of nitrogen fixation. So a completely new kind of process. These deep inside the nodule, there is a new signaling process going on that we've now discovered, and this raises the possibility that specific DNF1 processed peptides may be triggering differentiation. As Mergert and Kondo-Roshi have shown, these peptides are also able to carry out control of bacterial cell division and DNA endoduplication. So there are many hundreds of NCRs, and tracking down what each of them does is a big challenge for the future. From our standpoint, as we look at bacterial differentiation, we want to know, are these effects direct or indirect? Does an NCR peptide actually go into the plant, into the bacterium and interact with the receptor, or is it causing some condition in the plant that then causes gene transcription to change in the bacterium? So our purpose is right now to characterize the mechanism in the plant of how these DNF1 signal peptidase components are working, to ask whether there are other substrate proteins as well, and to find out their mechanism in symbiosis. So what I've shown you today is an outline of how we're taking a look at bacterial genes from the early nodulation processes all the way through the differentiation of the bacteria that makes it possible for them to fix nitrogen. And I think in particular that the use of plant mutants, as we did with DNF1, to look for plant factors that control bacteria may be an especially promising direction for the future. There are going to be many levels of regulation, from external signals to sigma factors. I think that the combination of genetics and in vitro transcription studies are important, and a lot of our effort in the future is going to be directed towards this double study of plant mutants and bacterial genes in order to try to dissect out the signals and the controls. So with that, I'll conclude this part of the lecture, and my third lecture is going to be on how the plant responds to the bacterial signals at the various stages of nodulation. Thank you.